We have Dr. Arun Mistry, who's from Minoryx today, who's here to give us, I believe, an update on some of the clinical trials for the Nexus study and information on the ongoing Calyx study. So, Arun, thank you very much, and take it away.
Thank you very much, Keith, and thank you very much to ALD Connect for inviting us to provide this update and also for all the support that you give to helping us with the clinical trials. I'm Arun Mistry. I'm the Chief Medical Officer at Minoryx. I've been there for 3 years. What I'd like to explain is who we are. We're a small biotech company based in Spain. I'm not Spanish, I'm English. But we're a small company, but we've got a lot of activity going on specifically in the ALD field. And we're looking at indications and a therapy for problems with rare CNS diseases which have a significant unmet need. We have one particular product, as I've said, which is called liraglutide. It's a PPAR gamma agonist, and it has several modes of action, which I'll come to. But in terms of where we are with this product and our development program, so we actually have for XALD, for ALD, a submission in with the EMA for an approval. We have a phase 3 study ongoing, which I think you've heard about, called Calyx. It's a global study, but approved by the FDA in adults with CALD, and I'll explain a little bit more about that. But also, as I've said, we're looking at other rare indications as well with the product, and we have a phase 2A study in Rett syndrome, which is another rare genetic disease in girls, young girls with neurodegeneration, and that's ongoing at the moment, and several other disease areas where we could look at, but our focus at the moment is in ALD and Rett syndrome. And just coming back to liraglutide, what is it? It's a PPAR gamma agonist. It actually comes from a family of drugs which have been used previously in and used currently in type 2 diabetes. However, this has a particular brain penetration profile, so is achieving significant levels of drug concentration in the CNS. And with that, we actually have activity in the CNS, which is anti-inflammatory, so reducing neuroinflammation. Actually, an activity on myelination in terms of reducing demyelination, and also some levels of remyelination as well. And then additionally, in many CNS diseases, there's a downgrading of the actual energy activity of the cells and the neurons. And what PPAR gamma agonists do, and particularly liraglutide, is upscale that with an action on the mitochondria, which are the sort of energy factories of the cell. And what all of this can do with all of these multiple modes of action, but particularly the anti-inflammatory actions, is help with neuroprotection, so protecting the brain from further deterioration. So we've been through what is ALD, what is CALD, and AMN, but just a quick recap, and just to say that our focus at the moment with our development program is around CALD, so the cerebral form of the disease. And this affects boys and men. We have seen examples today of also women being affected as well, but that's highly unusual. But when the cerebral form occurs, it leads to rapid neurological decline and death, quite often within 3 to 4 years. The other phenotype is adrenomyeloneuropathy, and this affects both men and women from adulthood, and I think you've seen the information on that. Also, that's focused on demyelination in the corticospinal tracts and the spinal cord. But we won't be talking about this. But both of these phenotypes can occur independently and coexist as well, which is again important. So focusing on cerebral ALD, through the course of the lifetime, and I think you heard through the presentations earlier this afternoon, quite a significant majority of boys and men affected by ALD will be affected throughout their lifetime by the cerebral form of the disease, up to 50% of the population. So a third of that occurs during childhood, and then another third occurs during adulthood. So that, again, is quite a significant risk that can occur. There are no prognostic indicators yet, but exciting research from the lipidomics and other sort of biomarkers that are being looked at the moment. Currently, there are two forms of treatment, transplant or Skysona, which is a gene therapy, but there are no treatments in a sense to prevent CALD. Of course, we've heard about the issues and problems with transplants not going well, and they are invasive. There's myeloablation, there's morbidities and mortality associated with them. They're not always widely available, difficulties in finding a donor. And in adults, there's a poorer survival because there's also additional comorbidities, typically from the adrenal-myeloneuropathy, which can make the patients much more fragile. Only in the US do we have gene therapy. In Europe, it's not available. The rest of the world, it's not available either. So patients who cannot have a donor and cannot have a transplant really are left with no other choices at the moment. So this is a figure that you'll see on several other slides coming forwards. So really just sort of thinking about how do lesions evolve in CALD. And I think we've been hearing about the work that Dr. Malak has presented with Dr. Mertz about there are areas which, in a sense, may not seem to have lesions, but there's probably something going on already with myelin content being reduced, or there's issues with vascular function. So that's probably a sort of starting point where on a typical MRI scan, no visible lesions are seen. Then lesions can appear. Typically, they'll start as gadolinium negative, which means that the blood-brain barrier is not broken. Then the lesions become gadolinium positive. So the blood-brain barrier is broken, that inflammatory process is started, and from what we've been hearing today is often self-perpetuating and very rapid. And that then means that the lesions grow, there's associated neurological decline, and then death over 3 to 4 years. These lesions grow before there are any clinical symptoms. So finding the lesions early with regular monitoring with MRI scans is very important, which I think you've all heard about again today, which is really important for a takeaway message. So if we put MRI scans onto this flow of how the lesions develop, you can start seeing that, in a sense, there's no lesions on this scan, but there could be in the background problems going on with the white matter outside of that. A gadolinium negative lesion in that second column that you can see, but then that changes to a gadolinium positive lesion, starts to grow rapidly, and then grows very rapidly, leading to neurological decline. You see that the biomarkers, like the scoring system called the LESS score, which we've not really talked about today, but it's a scoring system up to 0 to 34, capturing how lesions are growing. So that, together with the lesion volume, which is, again, very important. You see how the lesions are growing on these slides, change. There are biomarkers like the neurofilament light chain and new biomarkers that we're starting to hear about during the course of this meeting today. But all of these biomarkers and MRI changes, as I've said, precede clinical changes. So the worsening of the neurological status and the development of an increasing neurological functional score or major functional deficits like blindness, swallowing problems, mobility, they happen later. So in a sense, these lesions can be silent until these clinical problems happen. So really important to get the regular monitoring with the MRI scans. So what I'm going to take you through is some of the data that we have and new data which came through earlier this year, particularly in pediatrics, from the Nexus study. So I'll take you through that. This was an open label study conducted in Europe, but we have data that's been presented at AAN earlier this year. And we've met the primary endpoint, which is quite important for how regulators can see this data. And then also through a small amount of information on the advanced study, focusing again on the cerebral component of the disease. This was a very large clinical trial in adult men with AMN symptoms, but also we were looking at the cerebral part of the disease with regular MRI scans and also collecting information on the development of progressive disease. And then finally, we have a compassionate use program ongoing in Europe where men and boys are receiving compassionate use liraglutide. And some of the centers, and one particular center in Paris, has collected data in a sort of study-like setting and has published a paper which I'll take you through as well. So the Nexus study. This is the study in pediatrics. It's a study in boys aged 2 to 12 years, enrolling patients who are both gadolinium negative and gadolinium positive, with a LES score of less than 10, but with lesions. So we want patients who have been diagnosed with lesions on MRI scans, but with a LES score of less than 10, and also a neurological functional score of less than or equal to 1, so in a sense, neurologically intact and well. This was a study developed and designed as an open label study, so all patients were receiving the drug, and designed so that patients came into the study prior to transplant. So in a sense, patients were being diagnosed and could come into the study awaiting transplant or, in a sense, potentially avoiding transplant. The study was set up for 96 weeks, but we had an initial 24-week interim analysis, or a look at the data to make sure that, first of all, that the study could continue, that there was no concern that there was worsening of the disease with the treatment, and that was passed. So we went all the way through to 96 weeks. The primary endpoint, which is what is the question we're trying to answer, was about looking at the percentage of patients who had what we called disease arrest, both clinical and radiological. So a stabilization and stopping any worsening of clinical status, so making sure that neurologically the patients were well, and also radiologically stabilizing the lesions, not seeing any worsening of the lesions in any form as well. We also looked at other secondary endpoints, of course, like the LES score, the neurological functional score by itself, the gadolinium intensity score, and importantly, one of the additional areas I'll show you is around the lesion volume. So again, we have the LES score, but lesion volume is also important. You saw how the lesion volume increases quite rapidly, so we can now measure that quite clearly. And this study is really sort of now being positioned and has been submitted to the EMA for the primary data set for an approval. So you see the diagram again with the top row of how the lesions progress. And you can sort of see how what would be expected, in a sense, from that middle row in natural history. So the natural history of the disease is, as we've said, patients develop a gadolinium-negative lesion or a gadolinium-positive lesion latterly. The expectation from literature and several publications is that 10% of the patients who develop and are diagnosed with the lesion in the ages between 2 and 12 would have self-arrest. So that's the expectation, that 10% would self-arrest. What we saw with the study, with Nexus, we had 9 patients enrolled who were gadolinium negative, 14 patients enrolled who were gadolinium positive. And what we saw following them for 96 weeks, or up to the time point when they could escape to transplant, is that we had a 35% arrest rate, which was much greater than the expected arrest rate from just natural history. So we met the primary endpoint, and we had 7 out of 20 patients who were evaluable with the appropriate data set to assess from baseline to week 96, that arrested, which is much greater than, as I said, than the self-arrest rate. So we met the primary endpoint, which is important. But when we go further into the data and we look, as I've said, the volumetrics is quite a novel but important aspect of the disease because the lesions grow so rapidly, is that we saw, as you can see from this figure, the natural history growth, and this is on a log scale, so if you go back to our school days of mathematics, these lesions are growing very rapidly. On a log scale, we can get a straight line. And this gray area is where the lesions are seen to be growing as part of natural history. But the blue area is what we get from the study. So we actually get a differentiation from natural history with treatment on liraglutide. So the lesions are slowing down. And what we can say when we sort of look at the data is that we're seeing lesions growing around 4 times less slowly than natural history. So we're reducing the lesional growth from that anti-inflammatory effect. All of these data for the volumetrics are, again, correlated with the biomarkers, so the neurofilament light chain and also the MMP9, which is a breakdown of the blood-brain barrier, actually correlate very well with the volumetric data. And then when we look into the clinical and other radiological aspects, first of all, clinically, the furthest on the right, that's the neurological functional score. And you can see all patients, and this is all 23 patients on that figure, really remain clinically stable up until the time that they were on study drug. And we have patients now coming up to 48 months, 4 years in the study, still remaining clinically stable. What you see on the first 2 figures are the LESS score, so the radiological measurements that are taken, and split by the gadolinium negative patients first. So you see all 9 patients on that figure. You do see some patients going up and then stopping at about 12 to 18 months. These patients went to transplant. 3 patients from that group, 3 out of the 9, went to transplant. The other 6 have remained on study drug and are remaining stable with their LESS score. The middle figure is for the gadolinium positive patients. You can see that several of them follow the rapid growth and increase in the LESS score, but quite a lot of them show an increase and then a plateauing. And again, there's different ways of thinking about how the product is avoiding that growth. Of course, these patients are often remaining gadolinium positive, but the lesions are not growing as quickly as it would be expected. And then quickly onto the advanced study. As I've said, this was a large study looking at AMN symptoms. So this was, again, going back to the 6-minute walk test. we were unfortunately not successful on that, but when we look at patients who developed progressive CALD over a 2-year period, and this was a large study with 116 patients randomized, a 2-to-1 randomization, so 77 patients on active liraglutide, 39 patients on placebo. And what we saw over the 2-year period is only placebo patients, 6 out of 39, developed progressive CALD, which is a safety signal, but captured very clearly by multiple centers across the globe individually to say that these patients were developing progressive CALD. No patients on liraglutide over that 2-year period, so 0 out of 77, developed progressive CALD. So again, we can start seeing a sort of protective effect that the product may be having. And then with this compassionate use program, this has been used in a compassionate early access type of program in Europe for about 3-4 years now. In Paris, the data has been collected much more formally within an ethically approved data collection exercise. And what has been published in a paper in a journal called Brain is for a data set of adult men aged 19 to 67. And what was seen there is over a 2-year period, 10 out of the 13 patients actually remained clinically well. And these were patients who were diagnosed with gadolinium positive and also gadolinium negative, but progressive CALD. So they were diagnosed with clinical symptoms which were starting to show, and also clinically relevant rapid growth of their lesions. But 10 out of the 13 were clinically and radiologically stable for up to 2 years. 4 out of the 10 were stable with some level of clinical improvement. So the proprioception and neurocognition, so cognitive effects, signs were improving in these patients. 5 of the 13 patients actually came in awaiting transplant, and by the end of that 2-year period, were actually taken off the transplant list at that time point. So again, it's not a formal clinical study, but a real-world data set, which is really important to understand. So with that, just sort of need to talk about the safety of the product. And with all of the clinical trials that we've had ongoing, we actually have, for a rare disease, quite a significant safety data set now. So we have over 450 years of patient use of liraglutide in adults. We have over 100 years of patient years exposure in pediatrics. What we would expect to see from a PPAR gamma agonist from the history of the product being used in type 2 diabetes is peripheral edema, so fluid retention, and also some weight gain, which we did see much more frequently in adults. Typically, the adult men who had AMN symptoms were less mobile. In pediatrics, we saw some of the edema, typically facial edema rather than peripheral edema. The boys are quite active and running around. Weight gain was seen in both sets of patients, but weight gain which increased for 6 months and then plateaued out. The boys, yes, of course, they will continue growing, but they sort of follow a slightly different trajectory on their growth curves. With the fluid retention, we're not seeing any cardiovascular effects. So with fluid retention, one of the signals that was seen in type 2 diabetes, a much more older population, is that they would tip them into heart failure. We don't see any of that with the monitoring that we're doing, but we continue to watch that closely. So again, we're really reassured. We have a significant data set for safety, and there are no issues with the product crossing the blood-brain barrier, causing any other negative neurological symptoms either. And so now just to finish on the Calyx study, which you've sort of heard about in the past. You'll receive a flyer on the tables tomorrow morning just to take away. This is a study in adult men with CALD. And it's men with gadolinium-positive disease. And this is a study discussed and agreed with the FDA. So this is our pathway to a US approval. The study is enrolling men aged over 18 with gadolinium-positive disease. We're looking for 40 men, and those men who are not recommended for transplant, so either ineligible or not recommended or not wanting to go to transplant. We have sites open globally, so I'll take you over that. One of the other things that you've been hearing about today is natural history. Very important to understand how the disease progresses through a patient's, through the community and through a patient's life and the expectations of what would happen. For adults with CALD, there is very, very little data published, taken down. So one of the things that the regulators, the FDA, said, we don't understand the natural history. What you've presented isn't enough for us, so this needs to be a placebo-controlled study. So from the 40 men, half will receive active drug and half will receive placebo. And the study is designed as a survival study, which is unfortunate, but it seems to be the only way we can get an answer quickly before a longer data set to try and get natural history will be achieved. We are looking at other secondary endpoints of the LESS score, of course, looking at regular MRI monitoring. So this is the study design. And the study is designed with half the patients, 20 patients receiving active, 20 patients receiving placebo after randomization. But we have the opportunity and agreed with the FDA of looking at the data at 18 months after the last patient to see if we have a positive signal on efficacy of an outcome. If that's successful, all patients will transition to active drug into the open label phase. If that's not successful, we can go on for another 9 months and then again look at the data. This is all reviewed by what is called a data monitoring committee, so Minoryx won't see this data unless it's positive. So at 27 months, this can happen again. If efficacy is seen, then all patients transition to open label. If not, we go all the way through to 3 years, 36 months. At each of those interim time points, we actually don't have to increase the size of the study, which is the typical. If you're looking at data more frequently, often you have to bring more patients in. But the FDA has agreed that we do not need to increase the size of the study from 40 patients in total. And in terms of the key inclusion criteria and exclusion criteria, I've sort of mentioned them already. We're looking for adult men, gadolinium-positive disease, a LESS score of greater than 0.5, but less than or equal to 12. So in a sense, not with a very large lesion load, but still, in a sense, not recommended for transplant. So patients who are not recommended for transplant, in a sense, assessed to be having some level of advanced ALD disease and cerebral disease, but still early in terms of lesion load with the LESS score. We do allow patients in who have AMN symptoms of having mobility issues, being wheelchair bound, or having urological or bowel problems with incontinence, because these are issues from the AMN symptomatology. So they are allowed in. In a sense, the only key exclusions are previous transplant or gene therapy. Everything else, in a sense, should be fairly straightforward. So the study is ongoing globally. So first of all, in the US, we have Baltimore, KKI, Boston here, also Minnesota and Stanford, so centers you've all been hearing from today, but also centers in Brazil and Argentina. And then in Europe, we've set up the study during the course of this year. So the UK, France, Spain, Germany, and then also additionally a site in India, in New Delhi as well. So we do need the support of the community. We're about 2/3 of the way through, if not a little bit more than that, through enrollment. It is a difficult study. We know it's hard for families to think about, patients to think about, but please speak to the doctors in the room and understand how this study can help yourselves, your families, and future generations as well. You'll see this flyer tomorrow. There is a QR code. We do have IRB, so Ethics Committee approved pre-screening activities in the US where patients can attend for an MRI free of charge and also receive a stipend of $500 and also get a genetic test. So if these things need to be done, there are no costs to the patient. Also, it just improves routine monitoring. So capturing disease early, if patients are identified who could go for transplant, fantastic. I think that's one of the key things that the study is driving, is hopefully the awareness of routine MRI monitoring. Thank you very much.
Okay, we're over time, but I do want to allow questions for Arun, the Minoryx team here. I think this is an important topic. Are there any questions, Arun?
Hi, thanks for your work on this. How do you envision, if this is approved for cerebral ALD, how do you envision deciding how long somebody would need to take it? So obviously, if the lesions are still growing, they'll keep taking it. But if a patient appears to have arrested disease, do you have an idea of whether they just continue to take it for the rest of their life, or would it just be a shorter course of treatment?
So the data that we're collecting is longer-term data, especially in the pediatric population, where we're up to 4 years and longer for some of the patients. We know from the data that we've collected also for the adult study that even when patients came in with lesions at baseline, or no lesions at baseline, there was a chance of progression if they were on the placebo. So the development of lesions, the progression of lesions can happen at any time. So we will keep collecting longer-term data. We're unlikely to put a limit on the time for treatment. It's something that is a discussion between the patient and the physician. But in a sense, to avoid progression of lesions, to avoid the development of progressive CALD, we would sort of, from the data at the moment, is saying the product should be continued.